Thanks everyone, and thanks for the opportunity to speak today. I'm going to give some research updates on two projects and then a more detailed overview of the project that I'm leading in this program. So all of the projects that I'm talking about fall under the broad umbrella of looking at understanding the soil, plant and temporal mechanisms that influence the water that's available to crops in dryland systems. The first study that I'm giving a research update is that the one led by Terry Rose out of Southern University, and they've got two sites where they're looking at the influence of fallow cover crops on crop water availability to the subsequent cash crops. And they've been looking over the last 4 years at those two sites with a few different combinations, a couple of different cover crop levels, so 3 species mix and 9 species mix. And the site that I'm going to be talking about is the one at Burramine in northern Victoria, which is where our project is co-located as well. So I'll be talking more about that site as well. So Terry presented some results last year, and the additive results from this year, presenting a little bit on the charts on the right there. So on the y-axis we've got the ratio of the treatments, so the cover crop treatment plots, water that's available in the profile at sowing of the subsequent cash crop, the ratio of that to the control. So the control is a canola wheat rotation with no cover crop in the summer. So the one-to-one line is the green line going across, and so anything above that means there was more water at sowing than in the control, and anything below the line means there's less water. And there's a general downwards trend where the more summer cover crop biomass that was grown in that particular year, the less water available at sowing in the profile, which was what we might have expected. The biomass ranges are not particularly large. The summer cover crop opportunities were small in that environment to grow a lot of biomass. But the graph below, it's another ratio graph, but on the y-axis is the yield ratio, so the yield of the cash crop in the treatments that had a cover crop relative to the fallow control. And they're about equal number of points above and below the line, and that's related to what was available in the water, the soil water at sowing. So there was enough in-season rainfall to sort of normalise any effects of a deficit of water at the beginning of those seasons, which meant there weren't any particular significant anyway yield penalties observable in those three replicate study treatment plots. So overall there was that compensation but the overall suggestion is that the cover cropping in that environment is an opportunistic practice. It's not going to work every year, and you might see some water penalties, and you might have to rely on that in-season rainfall, which might not always be what you want to do. But we will come back to that with some interesting mechanistic information that we've discovered as well. The second fallow cover crop study is one being done by PhD candidate Han Lu Zhong, out of the University of Southern Queensland, and also supported by Queensland Government, and that is looking in the summer dominant rainfall zone with both summer or winter cover crops in 2 different years of study. And her studies in southeast Queensland, the axes are the same on this graph. We've got a lot higher biomass totals, up to 20 tonnes per hectare on the x-axis there in the top graph. Han Lu also presented last year, and that conclusion was that for a summer cover crop, that if that was terminated early, there was a good yield advantage in the subsequent wheat crop. And some of the other treatments with mid and late termination were also successful. And then flipping that to do a winter cover crop, followed by a summer cash crop of sorghum, the water profile at the planting was not related to the winter cover crop biomass. So in those red symbols in the top graph, we don't see that downward trend in the way we saw it at Terry's site, or in the way we see with the blue dots from last year in Han Lu's site. And again, there was looked like there was some above and below the line yield ratios in the lower graph. So again, some improvement, some non-improvement, and those differences were not significant in that year in Hanlu's study. So a range of responses, and what we have looked at in our study is trying to get a better handle on the mechanisms that are controlling some of those variations. The more we know about what's controlling the water that's available to plants, the better handle we can have on deciding what might be the best way to manipulate that. Is it worth manipulating that, and can it be done, and what difference would it make? So our project is based on the premise that the flow of water through the soil, the plant and the atmosphere continuum is controlled by three large concepts or factors. One being a capacity factor, how big is the bucket for the water. A second one being resistance factors, how much resistance is there to flow of that water through the system. And the third one being what is the potential gradient, what's the environment providing in terms of a driving drive for that water to move. So these factors and the mechanisms that link them, we believe need better description in order for all the models we want to be able to predict what will happen in one place, in one soil, with one management scenario in mind, and a suite of constraints underfoot. So that's where we're using the water retention curve, which is stylised here. That's the relationship between the soil water content, so how much water is in the soil, related to how tightly that water is held on the x-axis, the matrix potential. And if we can manipulate the structure of the soil and the way the soil is behaving and how the plants are reacting to that, then we can potentially improve water use efficiencies and yields. So our project is a large team across 6 organisations whose logos are there, and we've got collaborators in 2 farmers at 2 sites. We're also collaborating with the University of New England team up at one of our sites, and the Terry Rose team at a site. And there have also been a number of contributors prior to the beginning of the project and in the early stages of the project, and I acknowledge everybody because it's been a fantastic collaboration in the true sense. The research methods that we are using are a novel in-field approach to describe the soil water retention curve. So typically or conventionally, traditionally, the methods that we use to do that are taking an intact soil core to the lab, putting it on suction plates and having a look at the relationship between soil water content and suction. There are some in-field techniques, pouring a lot of water on a plot of soil at different contents, water contents, measuring those matrix potentials. What we're using is paired soil water content sensors and matrix potential sensors in the field. So putting them in the field so we can have them in there and have the plant in the system at the same time, and look at the changes over time, not just a static soil sample from one point in time from one smaller space. Sensors still have a restriction in how much space they sense, but a little bit more than a core. We've got 3 sites with 3 different ranges of treatments. We've come in to existing projects and looking at the treatments that they have imposed and trying to have a look at whether there are medium-term impacts of those treatments on the plant available water capacity and why. At the top, the brown soda sol in the northern Victorian Barramine site with the fallow cover crops is what I'll be talking about. Those crops have been in since 2019. We're also looking at a deep rip and lime gypsum trial up in Armatree, northern New South Wales with UNE. Again, treatments since 2019. And the third site we're looking at to see whether we can see any effects of a trial from 36 years ago out at Condo where there was some surface lime and gypsum applied. So back to the brown soda sol in northern Victoria and Barramine. These are our 3 methods that we're looking at. So the yellow dots on this water retention curve on the left are yellow squares are the data from our paired in-field sensors. We've overlaid them with data from cores that we've taken to the lab using HYPROP and WP4C instruments to measure the matrix potential and relationship. Those are the red, the blue and the green lines. And we've also used a filter paper method again on those intact cores and those are the black symbols. And interestingly, they overlay. We've got some agreement and alignment between the three methods. We didn't necessarily expect that. That's what we have in the surface soil. The subsoil on the right, we didn't get full alignment between the two methods of the benchtop methods, and we're still working on the data to plot the yellow dots, so the mystery will unfold as to where they sit. But what we are finding from that left-hand curve and alignment is to say, okay, we've got some alignment, let's go and use these infilled water retention curves to match up with what the plant's doing, the water use over the season, and even have a look at whether that water retention curve changes throughout the season and different drying cycles throughout the season. So some of the results that we've come up with, this is a time series of the total water available in the subsoil. So we've gone 25 to 75 centimetres in this chart, because the 75 centimetres was where we were largely seeing water extraction, the depth of water extraction. And this is showing from when we started our sensing, which was just before the wheat crop last year was sown, through to the red line there shows you the summer cover crop treatment. The blue line is the control, so no summer cover crop. And then now it's in canola this year, so the season's still going. And when we had a look at that, it all started off very wet. This has had 3 summers of cover crop prior to us being there, but immediately prior there was no cover crop. It skipped a year because it wasn't able to be established. And I'm pointing at that screen, but on the so we started off wet with both treatments, and then when we got a dry down cycle, you can see the first green circle there, ellipse. There was a wetting event. So the cover crop treatment used more water. It took more water out of the profile than the fallow control. Why is that so? I mean, that was we were not expecting to see that any sort of difference there, because the cover crop biomasses hadn't been particularly large at that site. But we are seeing something in this water use pattern. Not only that, but then when it did rain, it refilled to a greater extent. So we're seeing a larger bucket here relative to the blue bucket there. And then similarly, a greater drawdown on the water in that summer cover crop treatment plots relative to the fallow control at the back end of the season. And then after harvest, we had another wetting event, so the second green circle, and again, a greater jump for the cover crop treatment, taking in more water than the fallow cover crop. The fallow treatment. And then through that fallow period, we've got what we would expect to see, which is use of water by the cover crop and no water being used by the control. And then into the canola this year, we've got our third wetting event that we're looking at here. Again, more than double the water intake into that subsoil in that wetting event relative to the control. So we're very interested to see that, and we're very interested to relate that back to our studies in the water retention curve space to look at what is driving that. Have we got some changes in pore size distribution? Are there changes in water accessibility into that subsoil zone? We did also look at the rooting depths. Have I got that? I don't know if that's hidden or not. In that system. This was at the end of the wheat crop last year, so in October last year, where we looked at the control versus the 3 species cover crop treatment and the root counts in the upper subsoil, so at 25 centimetres depth, and at the deeper subsoil in 85 centimetres, which was probably almost beyond the limit of where the roots were getting to in both treatments. But we won't see significant differences between those, but we'll have a closer look hopefully at the root activity to help draw some lines of evidence as to what is causing some of those differences. So implications being that some cumulative impacts of fallow cover cropping on the soil water storage might be measurable in these systems. The water retention curves that we characterised were similar across the methods that we tested in that A horizon in Barramine. So we're going to do the same. We've got the same set of data for the Armatree site, and we'll be collecting the same for the Condobolin site. So we'll have a look at how well those methods align, how transferable. Can we use those methods as a bit of a protocol for looking to getting more mechanistic information and trying to describe what's happening with plant available water in the field to inform models and to inform decisions around what might happen if we do X, Y or Z to ameliorate a soil constraint. Thanks.